chapter 20? It's the last chapter before the test. Test is Thursday, the day of Thor.
Are we doing a lab before the test? Is that why there's...
Yes. There's a lab tomorrow.
We have so many.
Looking at mitosis.
We have like
Wednesday is biography and Thursday's test. Okay? So if you have so many tests coming up, you should probably study for this one now. So DNA replication, that's talking about when DNA copies itself. When does DNA copy itself? 10 points. If you can tell me in the cell cycle, when does DNA copy itself? I'm going to draw the cell cycle up here on the board.
Put phase after that? I'm going to wait until it draws.
I'm gonna spell that. What?
Do we have to put phase after it?
Nope. When does DNA copy itself? That's what this chapter is about.
Gotcha.
I'll give you a hint. It's one of these answers. When does DNA copy itself? White correct.
I'm not.
Peach correct.
Wait, on what?
Green correct.
I'd go.
Purple incorrect.
Are we purple?
Orange correct. And blue correct. Okay, so it happens during S phase. That's when the DNA copies itself. That's what this chapter is all about, DNA copying itself. Are you excited about the DNA copying itself?
Yes, sir.
All right, so guess what? I have some DNA right here. See that, Caitlin? You with me up here?
Sugars and phosphates.
This is DNA. DNA is made up of these things. These things are called what? Just shout it out. We already studied this.
Sugar,
nitrogen
base pairs.
Nucleotide.
Yes. Is that Blake? That's a nucleotide. A nucleotide is made of 3 parts. A sugar, a phosphate, and a nitrogen base. Remember that? We already studied that. The nitrogen bases come in 4 different types. 10 points if you can tell me the 4 different types of nitrogen bases found in DNA.
Do we have to do the whole letter?
Yeah, you can just tell me the letters. There's 4 types of nitrogen bases in DNA. In other words, X can vary.
Can we just
Sometimes it's shaped like that, sometimes it's shaped like that, sometimes it's shaped like
You
that,
have to put the name.
and sometimes it's shaped like that.
Just the letters.
You can put the letters or the names. Peach, correct. Why did Peach give me 2 pieces? That was weird. Blue, correct. Purple, correct. White, correct. Green, correct.
Seems like slanted.
Orange, correct. So the 4 types are adenine, guanine, thymine, and cytosine. The adenine and guanine nucleotides are known as purines. The cytosine and thymine nucleotides are known as pyrimidines. Do y'all remember that? People from Georgia are pure. Remember that? We've done this before, haven't we?
So the pyridamines, it was in the chapter, I remember that. But
Yeah.
are they called pyridamines?
They're called, so the purines are guanine and adenine.
In any order?
Yep. And the pyrimidines are cytosine and thymine. And it always happens where a purine bonds to a pyrimidine. See, the purines are bigger and the pyrimidines are smaller. And so when you make a DNA ladder like this is, you have a big one bonded to a smaller one. So it takes up the right space in the middle there. The middle can't be too wide or too narrow. It has to be the right size. So if you take a big purine and bond it to a small pyrimidine, you get the right size in the middle of the chain. So G always bonds to C, and A always bonds to T. I always remember grilled cheese. I like to eat grilled cheese. That was one of my favorites. G bonds to C, and A bonds to T. At, you can remember at. Grilled cheese to me is funner to remember. More fun. So this is all review about the structure of DNA. There's what adenine really looks like. Sugar, phosphate, nitrogen base. There's what thymine looks like. Sugar, phosphate, nitrogen base. Anyone remember what we call the sugar in DNA? Has a name.
Glucose.
Nope. First one to yell it out gets 2 points.
Deoxyribose?
Yes!
Everyone's waiting on it.
Deoxyribose is the name of the sugar in DNA. What color y'all?
Orange?
Orange, you glad I didn't say banana?
Yes, sir.
You see how G and A are purines, T and C are pyrimidines. People from Georgia are pure. People from Connecticut are pyros. Doesn't make any sense.
So those are the
I'm just
different
making fun now.
bases that are bound together, or different, they're the different nucleotides,
That's right. G, A, T, C.
the purine and the pyrimidine.
That's right. Purines always bond with pyrimidines.
2 types of nucleotides then, correct?
That's right. The G and A are the bigger purines. The C and T are the smaller pyrimidines. This was all review that we did from the chapter before. Now what you need to understand is what the DNA is going to do when the DNA copies itself in S phase. What we have to do is we have to take this DNA double helix and we have to break it apart and copy each side. And we're going to do that with some special enzymes that you have to know. Some special enzymes that take part in this process. The first one's called DNA helicase. DNA helicase. I'll write them up here. DNA helicase. Guess what DNA helicase does? How could you guess? You don't know. It comes in here and it unzips the DNA. You want to see it unzip the DNA?
Sure.
Waverly, you watching this?
What?
Huh? Wasn't that good, Sam? Did you miss that?
No.
You seem like you were missing it. Let me do it again because that was fun to do. You ready? DNA helicase, here we go. Beep. It unzips the DNA. That's what that enzyme does. In reality, the enzymes are a lot bigger than this, but I've made them small. In reality, it would be like this, and it would lay over the DNA and open it up. Then another enzyme comes in called DNA primase. And what primase does is it attaches what's called an RNA primer to the DNA chain. DNA primase attaches an RNA primer. Now RNA, do y'all remember where RNA is found in the nucleus of a cell?
The nucleolus.
In the nucleolus. There's little pieces called RNA. They're like DNA, but they're RNA. They're a little bit different. They have ribose as the sugar instead of deoxyribose. So they have a different sugar, so they're called RNA instead of DNA. But what DNA primase does is it takes a little piece of RNA, just like this, and it attaches it to the DNA chain. Let me go get another one, it says, and I'll attach it. And this little piece of RNA needs to be on there for the rest of the process to work. And let me get a couple more of these. I'm going to attach over on this side too. Click. Oops, I want to attach this one first. Click. And click.
Do the enzymes?
Now both sides are primed. What's that?
The enzymes just leave afterwards.
Yeah, they just float away. They're actually just bouncing around everywhere in the cell, and if they happen to run into the DNA, they'll do their job. And if they don't, they won't. But there's a whole bunch of them, so they'll do their job. They'll get it done. Okay, that's DNA primase. Puts on a little bit of RNA onto the DNA strand. And then the next one is this one. It's called DNA polymerase.
It's a lot of base.
A lot of enzymes. Yeah, if you ever see ACE at the end of the word, that means it's an enzyme. All enzymes, or most enzymes, have ACE at the end. DNA polymerase, what does it do? It grabs DNA nucleotides that are floating around in the nucleus, and one by one, it attaches the DNA nucleotides to the RNA chain. Whoops, I dropped one. Attach, grab, and attach. Grab, and I grabbed the wrong one. Grab and attach. See how we're copying the DNA?
So the polymerase it copies it?
Copies the DNA. It takes the strand that's already there, and it adds on new nucleotides to the other side to get it copied.
And then the primase attaches primers?
The primase attaches this little RNA primer that we're going to remove later. Oh, and by the way, the polymerase does it on the other side too. Attach, grab, attach, grab, attach. I need one more. I don't have the right kind. Grab, attach.
So that.
See how we're making two copies? It's kind of like that picture up there. Only I'm going up instead of down. We started with one, and now we're getting two copies. Is this exciting? We're copying the DNA, y'all. I want you to notice something. This side right here is copied in the direction of the helicase's unzipping. So the helicase is moving this way down the DNA, and this side is being copied in that direction. We call this side the leading strand.
Does it get copied first?
No, that happens at the same time. Because there's many of these enzymes doing their job. The other side is called the lagging strand. And the lagging strand gets copied backwards. Where the zipper's going down this way, the helicase is going down this way, but the lagging strand gets copied upwards, away from the helicase.
Why is that?
And it's because DNA always has to be copied in the same direction. DNA can only be copied going this way. And do you notice that this side, my nucleotides are straight up, and this side they're upside down?
Yeah.
So it has to go in that direction. We call that the 5' to 3' direction. But that's something y'all don't have to worry about. That's what we talk about that in AP, how the sides are labeled.
Honors has to know that.
5' and 3'. Does honors have to know 5' and 3'?
Yep.
Oh, okay. Well, that's easy enough. The end with the phosphate is called the 5' end. See how that phosphate sticks up in the air?
Yep.
That's the 5' end. The end without the phosphate is the 3' end. So this side runs from 5' to 3'. This side runs from 3' to 5'.
So if it's sticking
For
up,
your honors guys.
if it's sticking up, it's 5. And if it's
Where
down-
the phosphate group is the 5' end.
Okay. Is that all we need to know for the 5'?
The new strand always goes in the 5' to 3' direction.
Wait,
Can you
what?
tell the phosphate is sticking out there?
Mm-hmm.
That's the 5', and so the new strand goes that way, and on this side the new strand goes that way in the 5' to 3' direction.
What is the 3?
The 3' is the opposite end. So if you look at a nucleotide, the 3' end is the top, the 5' end is the bottom.
So, you said the 5 is where the phosphate?
5 is the phosphate end. So that's the phosphate.
And the other one's sugar.
And the sugar is the 3
Oh,
end.
okay.
Is that all we have to know for that?
Yeah, that's all you got to know. So
So that's just where it
the
is.
3 and 5 are ways of saying if it's right side up or upside down.
Okay.
That's all it is.
But couldn't it be switched to left and right? If it was upside down, wouldn't it just be the same?
Yes,
Like the left side's always going to be...
this is 5 and that's 3.
So that's
This
how
is 3 and that's 5.
So the left side's always going to be 3
This
to
is
5.
5 and that's 3.
Wait, no,
Confused? Okay, so whichever side that's in, that's the 5' end. That's what we call it. That's what
So it has to be
scientists call it.
copies from 5 to 3.
So that's the 5' and that's the 3'. Okay, wait, I'm going to turn it upside down, watch. That's the 5', that's the 3'. 3', what is that? There you go, you got it. So if you look at this side right here, that's the 5' end, that's the 3' end. Because it's upside down. You look at this side here, that's the 3' end, that's the 5' end, because that's the phosphate. Does that make sense? Oh, now you got it. Anyway, the new strand that's copied always goes from 5' to 3', so it has to go in that direction. These numbers are ways of saying direction that scientists use. The numbers are actually the carbons that are on the sugar. Scientists number the carbons. It's a 5-carbon sugar. And so they number it 1, 2, 3, 4, 5. The 3 carbon is on top and the 5 carbon is on bottom. That's where the numbers come from. Nate, are you worried about the numbers? See? That's why I'm telling you. You good now? Good job. That's confusing. It's a lot to it. It's really not that bad if you erase all this stuff that I've drawn on it. What we do is we copy one side, we copy that, and then another special other type of DNA polymerase, this DNA polymerase again, is going to come in and remove the primers and put nucleotides in its spot. I'm out of nucleotides, but